All right, kids, I really want to take you on a field trip. And so the Science History Institute is right here in Philadelphia. So we're going to go inside. However, COVID, so I got to do this. And I can't go inside. We'll find another way. John Kendrew was the first scientist to come up with a 3D image of a protein. And the protein that he came up with an image for was myoglobin. He came up with this idea when he was in Sri Lanka during World War II. You see, a lot of scientists during World War II got placed as science advisors for the war, and he was one of those science advisors. And he's in Sri Lanka in the jungle talking about science and his chemistry research, what he wanted to do. And they said, it would be a good idea if you studied protein structure. And he said, okay, well maybe I'll do that. So the war ends, and so John Kendrew was from England, and when he's on his way back to England, he went by way of California, where he ran into Linus Pauling. And Linus Pauling said to him, you know what, X-ray crystallography might just be the ticket for you. So he came up with the idea of doing X-ray crystallography on myoglobin. 7 years later, he finds a viable source for myoglobin, which is sperm whale hearts. And so he manages to grow beautiful crystals of myoglobin, but he wants to come up with the structure of those beautiful crystals. So he spends years and years and years analyzing X-ray diffraction crystallography images to come up with the first image of a protein, myoglobin. But the image looked horrible. It looked like a brown snake, like coiled around some sort of like giant watermelon of sorts that was invisible, and it didn't really appeal too much to the masses. So in comes Irving Geis, and Irving Geis was an illustrator, and he comes up with this illustration of myoglobin. And as you can see, there's a metal center that is binding to oxygen in myoglobin, and he illustrates it in a way that makes the cover of Science magazine, and it's beautiful, and it transforms the way that people looked at science as a molecular study of the body. Here is not myoglobin, this is chymotrypsin, but this is the kind of model that that Kendrew would have made, so that he could draw so that Irving Geis could draw the illustration. All of these parts on here, all the little metal pieces and the different colored tips represent the different amino acids in the protein sequence. Okay, this is the story of Jenny Glusker. Jenny Glusker was a child of the kind of the way that women were oppressed back in the 40s, 50s, and 60s. She actually used that to her advantage. So when Jenny Glusker was a child, there wasn't a lot of women that were going into science, right? But then World War II broke out. And so she was able to go into science because all the boys went off to war, right? And so she was able to fill that role, right? So she went to college and she studied chemistry. And then while she was working on her PhD, she had the fortune of working with another woman in chemistry, Dorothy Hodgkin. Had to remember her name. Dorothy Hodgkin, while Jenny Glusker was getting her PhD, Dorothy Hodgkin was working on isolating the structure of vitamin B12. And she actually won a Nobel Prize for identifying the structure of vitamin B12, while Jenny Glusker was her PhD student. Then Jenny gets married, takes some time off from chemistry to have some kids. With her husband, she moves to the United States from Oxford, from England. And then they're looking for a lab where they can work. And then that's when she meets Arthur Lindo Patterson. And Patterson had a lab in Fox Chase here in Philly, and he was very eager to take women scientists and allow them to do real science, which was pretty revolutionary at the time. While Jenny Glusker was with him, she, using X-ray diffraction crystallography, which is what this machine does. This is the X-ray source for X-ray diffraction crystallography. While she was working with him, she isolated a lot of different drugs and chemicals and tumor-causing agents that were the sources of cancer. And so her work directly benefited many, many people who had cancer and then were able to recover because they could identify the specific structure of the tumors as the structure of the chemicals and the interactions with those tumors. So much of chemistry, as we know it today in medicine, are connected with spectroscopy, or the interaction of light and matter. So in 1919, Otto Folen used a tube like this in order to develop a blood glucose analyzer. And he would take a reaction of copper ions, copper 2+ ions, in an alkaline solution or in a basic solution with blood, and he would be able to measure the glucose level because glucose is a reducing sugar. And so it would change the color of the copper as the copper was reduced. And so they were able to analyze blood sugar back in the 1920s with that apparatus. Fast forward to 1973, and they developed a digital way of doing that by passing light through the sample and measuring the absorbance of the blue light. So how much of the light, the blue that was transmitted, depended upon how much blue color was in the solution, depended upon how much sugar was in the blood. So simple color change experiment was used to determine blood sugar levels in patients that were diabetic or pre-diabetic, or just to know what the blood sugar level was. So you may ask, why is Mr. Wolf such a huge fan of chemical demonstrations? Well, it goes back to the 1800s with Michael Faraday. In fact, that was the way in which chemistry was educated to the masses, was through public demonstrations. In fact, it would be a huge event for a scientist to put on public demonstrations of their chemistry knowledge and their chemistry abilities to teach people. And so this is an example of some of those things. They would get together and teach, and that would be the public event for the evening. It's before, when schooling was very expensive and it wasn't available to everybody, this is a great way to expand everyone's knowledge. So how do we know stuff? How do we measure things in our environment? Well, part of that is the instruments that we have. But back in the day, they didn't have instruments. If chemists wanted to learn something and explore something new, they had to invent a new instrument to do that. Well, James Lovelock's electron capture device is one of those examples. D6, he was looking for a way to get an instrument that was more sensitive. He was working with biological specimens, and he wanted to be able to detect things inside of cells. And with the things that were available at the time, that was impossible. But he had access to a huge warehouse and all the materials that he would need. So what he did was he invented a new electron capture device, which is based off a gas chromatograph. And so what it would do was you would send a sample through the column, it would separate out the sample based on its particle size. And then at the end, he would shoot electrons through the sample and it would charge the sample. He would be able to detect, based upon the current and the change in the current from the electrons moving, what particles were in that sample. And he could determine that at a level of sensitivity that was parts per billion or parts per trillion. He told the story to tell you about it, that someone could open a bottle of ether in Japan and he could measure that, he could detect it in England two weeks later. It was an amazing hypersensitive tool that we still use today, and it actually led to the environmental revolution that happened soon after in the 60s and 70s, because he was able to show quantitatively that humans had an effect on the environment that was measurable.